Hi everyone, my name is Sarah Norton and today I'll be talking about nutrition, longevity, and disease. Doctors and scientists are always trying to find a way to improve human health so that way that we can live longer. One way to do this is to look at the level, type, and timing of food consumption, and this is the most feasible way to help human health, which leads to longer lives. This can be seen as well in microorganisms. There is a relationship between the nutrients and the cellular components, and these nutrients, as we know, will help fuel numerous metabolic pathways. We must then adjust these interventions based on age, sex, genetics, and metabolic risks. There is also a link between nutrients, fasting, genes, and longevity in short-lived species like yeast and worms, and this can also be comparable to primates and humans. Within this part of the presentation, we will be looking at how different nutrient intakes for yeast, flies, and worms can affect their metabolic and growth regulatory pathways, and then this will allow us to look at the comparisons between these small organisms and larger organisms like humans. When looking at yeast, two types of lifespans were studied. First is chronological, which it means the study of the non-dividing cells, and then replicative is the study of replicative capacity of individual mother cells. In yeast, aging is assessed through survival tests or replicative capacity measures. Nutrients, particularly sugars and amino acids, regulate lifespan pathways. Glucose activates certain pathways while amino acids regulate others. Mutations decreasing pathway activity extend lifespan by enhancing stress resistance and reducing DNA damage. Recent studies show that caloric restriction, or CR, and dietary composition affect longevity pathways involving glycolysis, oxidative stress, and autophagy. Similar pathways influence longevity in the nematode. Dietary restriction extends lifespan by modulating growth, signaling, and stress response pathways. Fasting induces pathways involved in proteostasis and mitochondrial function. In both yeast and worms, changes in growth affect metabolism, influencing proteostasis and longevity. Reduced insulin-like signaling extends the fruit fly longevity, and it involves factors like AKT, and large-scale studies in flies allowed for detailed exploration of genetic and nutrient interactions. It can be seen through the metabolic hub like phosphoryl pyruvate, amino acids, and alpha-ketoglutarate are able to influence longevity, and then flies that fed on citrate or beta-hydroxybutyrate showed increased health and lifespan. Macronutrient balance affects survival, with extremes in protein levels showing negative effects. Dietary restriction, or DR, in flies activates oxidative metabolism, stress signaling, and autophagy, with differences depending on age and genetic background. Intermediate fasting and time-restricted feeding delay aging, preserving proteostasis and impacting stress response and inflammation pathways. Studies in short-lived species highlight the role of genes in regulating health and longevity, with diet influencing key aging pathways. Mechanisms associated with fasting, such as stress resistance and reliance on lipid fuel, are shared features of delayed aging. Cellular processes like mitochondrial metabolism likely play a causal role in longevity and is induced by dietary manipulation. To summarize, short-lived species offer crucial insights into nutrition and aging research. Genes significantly influence the health and lifespan of these organisms, with aging pathways regulated by nutrient levels. Simple organism studies have revealed that genetic roles are responsible for nutritional cues of longevity. Complex interactions exist between nutrient composition and longevity pathways, with diet efficacy at the center stage. In short-lived species, aging is regulated via growth inhibition and metabolic pathway alterations. Fasting-associated mechanisms like stress resistance or lipid fuel use can be seen, and they are pointed to activate proteostatic pathways to delay aging. Cellular processes such as mitochondrial metabolism, autophagy, and stress response like drive are diet influenced and can lead to longevity, which can be conserved in mammals. Within this section, we are going to explore the effects of specific nutrients on the genetic pathways that regulate aging and diseases within mammals. It has been seen that in diets of high proteins and specific amino acids within rodents, there has been a shortened lifespan because the growth hormone signaling and insulin-like growth factor have been increased, and this can be seen because when genetic mutations occur that cause these factors not to be present, the rodent will then live longer. And so if we look here, you can see that this is where growth hormone and IGF-1 are present. So if they are not suppressed, they can go to insulin, and then insulin resistance, which then can cause inflammation or metabolic dysfunction, which leads to aging. So it can be seen that these play a large role. So we do need those to be almost inhibited when no growth is needed. And then these mutations can occur via medicine as well, and that can be given to the mice and humans, so that way no insulin sensitivity and diabetes, any bad diseases like that can occur, and it causes the patient to decline. Sugars, like proteins, speed up aging by boosting insulin release and activating pro-aging pathways. Glucose triggers pathways and will activate transcription factors like the M1 pathway, which can be seen right here. And then this will activate transcription factors that can then lead to aging, like oxidative damage, which then can lead to neurodegradation, and that is not good for us, and that only makes our age and everything decline. And so by targeting pathways like IGF-1 or TOR, we are able to offer a potential for anti-aging drugs, and dietary interventions may be able to provide safer and coordinated responses against aging, unlike the drugs. If the person is already on a lot of medication for other conditions, they might be able to change their diet to make sure that they are not intaking so much sugar that it is causing this oxidative damage. And this is something that can be done if there are dietary restrictions or food scarcity, then the patient can then look for the medication to help with this. Caloric restriction, or CR, has well-documented benefits on health and longevity in rodents, primarily through nutrition-responsive signaling genes. CR leads to decreased adiposity and improved immune response in rodents. Within monkeys, long-term CR has proven to have similar benefits, including lower adiposity, improved glucose and insulin sensitivity, as well as delayed brain aging. Short-term CR has been used within humans, and it has showed effects on body composition, insulin sensitivity, and biomarkers of health. These findings suggest strong conservation in how CR impacts health across mammals, supporting its potential for improving longevity. The biology of mammalian caloric restriction involves conserved cellular processes like autophagy, proteostasis, and metabolic shifts towards lipid fuel usage. However, the efficacy of CR depends on factors like diet, onset timing, sex, and individual genetics and metabolism. Personalized dietary interventions are key to optimizing health and longevity. There are many different types of fasting that were presented within this research. First is intermediate fasting, which means that you are going to fast about 12 to 23 hours a day. And then for rodents, they use time restricted feeding, which improves your metabolic health but is not as effective as CR in delaying age. And then in humans, they use time restricted eating, which showed weight loss and improved metabolic health. But there are two types of fasting you can do within this, either 11 to 12 hour or 8 to 10-hour fasting, just whichever works better for your schedule. Next, we have periodic fasting and fasting mimicking diets, which are alternatives to intermediate fasting. Periodic fastings are where you will not eat for 2 to 4 days, but this can only be applied once or twice a month because it can become dangerous to not eat so many days in a row so constantly. That can cause you to have an eating disorder. For FMDs, these are going to be a low calorie, plant based diet that mimic water only diets, but provides more safety because you still are getting some sort of nutrients. The FMD cycles were able to express anti-inflammatory as well as metabolic and regenerative effects, making the potential for disease prevention and treatment much more present within both humans and rodents. For the first question, I would like you to describe the differences between caloric restriction and the following types of fasting: intermediate, periodic, and fasting mimicking diets. You should be able to find all the answers within the previous slides. In addition to fasting, diets have been provided within this paper so we can kind of see what is the best way to go about our nutrition. For instance, we have high calorie diets, which is going to be very high in saturated fats and sugar, and these contribute to obesity and metabolic dysfunction. Low carbohydrate and ketogenic diets are going to have limited carbs and high fat to moderate high proteins, which can show cognitive and metabolic benefits. But long term, we are not sure how these diets will turn out. For low protein diets, you're going to have a high amount of carbs, and this is associated with longevity in rodents with sex specific responses. And then for the high protein diet, you are going to have a high amount of mortality because it does not offer significant health benefits. But then plant-based diets are linked to reduced mortality and disease risk, although these diets may pose risk for bone health due to amino acid deficiencies. Overall, there must be a balance of macronutrients so the health span becomes as shaped as it can be. And this also needs to be personalized for each person based on their age, sex, genetics, and lifestyle. All of this has been leading up to the longevity diet, which, according to these researchers, are going to be the best way for you to live the longest and get the most amount of nutrition. So, first, you are going to have a mid to high carb intake. Then, you will have a low but sufficient protein intake. Third, you will have moderate fat consumption. Fourth, 12 to 13-hour daily fasting periods. 5, avoidance of malnourishment. 6, body composition guidelines like guidelines for your doctor or just different ways to stay in shape physically. And then 7th, you will have evidence-based support, which means that you will go to the doctor and they will say, hey, you've been doing a really good job and this is how we can kind of improve you from here. And so when this diet is used as a preventative measure, it can complement standard healthcare really well and will allow you to live much longer. For our last question, I would like you to describe the principle behind the longevity diet, and do you think that it can really work, and do you think that people will end up living longer? And here are all my resources for this presentation. I would just like to thank you all for listening.